1. Introduction to the thyroid. Anatomic origin. The thyroid is one of the largest endocrine glands, developing from the floor of the pharynx in early vertebrate evolution. Metabolic master. Its primary function involves the production of iodinated hormones that regulate basal metabolic rates and protein synthesis across species. 2. The endostyle connection. Evolutionary ancestry. The thyroid evolved from the endostyle, a filter-feeding organ found in primitive chordates like Amphioxus and sea squirts. Functional transition. During vertebrate evolution, this mucus-secreting structure transitioned into an endocrine gland capable of sequestering iodine from the environment. 3. Phylogenetic conservation. Universal presence. The thyroid gland is a defining characteristic of all vertebrates, from primitive jawless fish to complex mammals. Shared functionality across all these groups, the core purpose remains the regulation of growth, development, and energy homeostasis. 4. Hormone structure stability. Chemical identity. The hormones T3, triiodothyronine, and T4, thyroxine, are chemically identical in every vertebrate species studied to date. Ancient molecules. These molecules are so ancient that even some plants and algae contain iodinated compounds similar to vertebrate thyroid hormones. 5. Thyroglobulin, the giant protein. Storage scaffolding. Thyroglobulin is a large glycoprotein where thyroid hormones are synthesized and stored in the gland follicles. Sequence. Persistence. Significant portions of the thyroglobulin protein sequence are highly conserved, particularly around the specific tyrosine residues where iodination occurs. 6. The role of TSH and TRH. The control axis. The Hypothalamic-Pituitary-Thyroid, HPT, axis governs thyroid activity through thyroid-stimulating hormone, TSH. Receptor conservation. The structure of TSH receptors is remarkably similar in mammals and amphibians, allowing for cross-species hormonal activity. 7. Functional diversification. Metamorphosis control. In amphibians, thyroid hormones are the primary triggers for the dramatic transition from tadpole to adult frog. Thermogenesis. In birds and mammals, the thyroid took on the critical role of regulating endothermy and heat production in cold environments. 8 exceptions. Invertebrate analogs. Extrathyroidal iodine. While invertebrates lack a thyroid gland, many use the same biosynthetic pathways to incorporate iodine into proteins for structural purposes. Functional divergence. In these species, iodinated molecules often serve structural or signaling roles that are unrelated to the vertebrate metabolic definition. 9. Genetic sequence divergence. Deiodinase variations. While the hormones are the same, the enzymes that activate them, deiodinases, show varied sequences across different lineages. Adaptation speed. Gene sequences for hormone transport proteins have evolved more rapidly in some species to meet specific ecological demands. 10. Unique mammalian features. Follicular organization. Mammals possess a highly organized follicular structure that allows for massive storage of prohormones compared to fish. C cells and calcitonin. The mammalian thyroid also houses C cells for calcium regulation, which are separate organs in many fish and reptiles. 11. Thyroid health and environment. Environmental markers. Because the thyroid is so sensitive to iodine, it serves as a biological indicator for environmental health across different ecosystems. Anthropogenic impact. Pollutants like perchlorate can disrupt this ancient signaling pathway in both humans and aquatic wildlife alike. 12. Conclusion. A biological constant. Core continuity. Despite 500 million years of evolution, the basic molecular machinery of the thyroid has remained essentially unchanged. Future research studying the minor sequence exceptions in non-mammalian vertebrates helps us understand thyroid-related diseases in humans.